So let's get started with our program. We are tickled pink to have Gail Langellotto here tonight. For those of you who may not be familiar with Gail, she's a professor of horticulture at OSU. She has a BS in biology and an MS and PhD degrees in entomology, all from the University of Maryland. She completed her postdoc studies at UC Davis and was an assistant professor of biology at Fordham University prior to joining OSU in 2007. She worked on various agriculture and entomology projects in California, Hawaii, and New York before she came to Oregon. And as most of you know, she held the position of statewide master gardener coordinator for OSU Extension until last year. She did that, she told me, for 17 years. So she's done a great job. We were all excited to work with her. She's always an excellent speaker, and we are pleased to have her here tonight to talk about the Garden Ecology Lab at OSU, which BCMGA supports financially. So please welcome Gail.
Thank you, Lynn, and thank you. Yes, it's already recording. Thanks all of you for coming this evening. I was telling Lynn that since moving to the Willamette Valley over the years, I've developed like really bad allergies. I had them as a child, and I've had really bad asthma also. Then COVID happened and I couldn't do my allergy shots. I'm telling you this to say I have this weird pitch in my voice because my lungs are a little bit compromised. I've been tested. I don't have COVID or the flu, but occasionally I might have to take a break when I talk for long periods of time. So I apologize in advance and thank you for your understanding. When I knew I was going to come to speak to the Benton County Master Gardener Association, I know that many of you have seen my talks multiple times, and so I wanted to share something new with you this evening. This is only the second time I've ever given this talk. The data that I'm going to share with you this evening hasn't been published yet, but it's been submitted to Environmental Entomology for publication consideration. I'm going to be speaking on the work of my PhD student, Jen Hayes, by and large. As Lynn mentioned, I transitioned out of the Master Gardener program and into a research teaching position last year. And when that move came, I was like, well, our work is called the Garden Ecology Lab. And I always appreciated the strong link with Master Gardeners as a way to directly get the research that we're doing into the hands of people who could put it into practice. And when that position changed, I've been challenged to think, well, how am I going to keep that connection? Well, one way that I'm doing it is we developed a new lab website that I encourage you to check out. And specifically, I want to call attention to this For Gardeners section on our lab website. In the For Gardeners section, we have these 2-pagers where we take the research that we're doing and translating it into an accessible, hopefully visually interesting PDF. Tonight, I'm going to be talking a lot about native plants. And as I start getting into the talk, you'll see that how to identify what is actually a native plant at the point of purchase is not very easy to do unless you are a well-studied botanist. So these are just some examples of some of the front pages of the 2-page briefs. This is from Aaron Anderson's research where he looked at the native plants that most attracted natural enemies in general, specifically had a high diversity of parasitoids or a high diversity of predators. And then, oh, someone is mentioning that the slides are too small. Could I ask? Okay, it's in presenter mode on your end. So I will, how do I change that? See, I thought I had it totally. I will stop share. My apologies, folks. And then I will go here and I will stop the slideshow. And then I will go to slideshow.
At the very bottom, there's a slide that says 76%.
Yeah. I don't think that's it. I think, okay, that should take care of it, but now I need to go back and do my, I need to share.
Can't see us.
Everyone here, folks, are happy to see the 26 folks that are online. They're all saying hello. Okay. So this should be better. So folks online, is that a little bit better? Can you see the entire screen now? Okay, thank you for letting me know, and my apologies for not getting that correct. Okay, so it's just some examples. Oh, then I need to hide that.
Yeah, there you go.
Okay, I need to hide that. Just some examples of the briefs that we have published. If you want suggestions for native plants which attract parasitoids, predators, and a high abundance of natural enemies in general, you can find it on our website.
Oh, good call. I forgot about that.
This is a outcome of an undergraduate research project where Jen Hayes, the research I'm going to talk about tonight, really did a study on native plants versus nativars, native cultivars, and whether or not they attract pollinators in the same abundance and diversity. Well, one of the undergraduates noticed that there were differences in petal cuts on the wild type native versus the nativars. So she summarized her research in another garden ecology lab brief. And just a note, as I mentioned, we do have information on how to identify a wild type native plant versus a cultivar blend versus a subspecies. So if you are wanting to know more about how to identify a native plant, how to distinguish it from a subspecies, variety, cultivar, hybrid cultivar, and ecotype, which could be valuable as you're getting ready for spring plant sale season, and these different plant types are going to be on the line ready for purchase. We also have an infographic about that. So please check it out, gardenecologylab.oregonstate.edu. I want to make sure that I get through the acknowledgements before I get into the talk itself, because I want to make sure that the folks who actually did this work get their due. You know, I turn 53 tomorrow. I find that occasionally I go out and I try to help them weed, but I like weed on my knees for like 2 minutes. And I'm like, well, I think I'm done for now. You guys look like you have it taken care of. So all of the students that you see here were folks who contributed to the research that I'm going to talk about tonight. In particular, I want to point out Jen Hayes, whose PhD work I'm going to spotlight in particular. But all of these students, past and present, have contributed to the work that you're going to see. I also want to give a shout out to Lincoln Best, who is the bee taxonomist with the Oregon Bee Project. Part of the reason why we're able to actually share some novel discoveries that other researchers haven't been able to do is because we had Link's expertise to identify bees to species so that we could see patterns that were precluded in other studies when they didn't have the ability to actually look at specific species. I also do want to thank the funders of our work. Our work is entirely funded by donors for the most part, a grant here and there, but donors including the Benton County Master Gardener Association help us do this work. I want to thank you, let you know that your contributions go directly to support these amazing students so that they can study and do good work and then go out into the world and do good things. I also do want to acknowledge that the research occurred at the Oak Creek Center for Urban Horticulture, the traditional homelands of the Marys River or Anthony Faux Band of the Kalapuya, which I think is particularly important since we're doing a project on native plants. Okay, I want to start out by giving a brief overview as to why native plant status may or may not matter to pollinators. And as a matter of fact, when Aaron Anderson first came to me and said that he wanted to study pollinator associations with Willamette Valley Pacific Northwest native plants, I thought, it doesn't matter. Being native doesn't matter to pollinators. And I want to tell you why. I do want to point out, though, that these are my 4 favorite native plants from Aaron's study of these associations. I put them up there because I think they're just such good eye candy. So we know that native plant status matters to herbivores, but pollinators are not herbivores. They're not herbivores in the sense that they don't chew on plant foliage the same way that caterpillars do. The reason that we know that native plant status matters to herbivores, such as caterpillars and other plant-feeding bugs, is that we know that for all of the insect herbivores that there are, about 70% of all insect herbivore species are a specialist. That means that they're only capable of feeding on plants within one taxonomic family. And the reason that people think that that relationship occurs is because plants are not sitting ducks. They are sessile, they cannot move. However, they put out a lot of chemical compounds which try to deter insect feeding. And because they're putting out so many nasty compounds, either feeding deterrents, digestibility reducers, straight toxins, only insects which have a long evolutionary associations with these plants have developed the adaptation so that they can effectively feed on these plants. In the case of monarch butterfly caterpillars, for example, milkweed has a lot of nasty toxins in it, cardiac glycosides, which would be toxic to any other insect. But what the monarch butterfly caterpillars do is they actually put that chemical, they store it in their exoskeleton, which is a biochemically inactive part of their body. It would be like us storing toxins in our hair or our fingernails. And because it's relatively biochemically inactive, the insect doesn't have to process that chemical in the same way. If you've read any of Doug Tallamy's recent works, starting with his really iconic tome, Bringing Nature Home, he does a really good job of putting forth an argument as to why native plants are so critically important to insect herbivores, which are 50% of all insect species. Just a couple of slides from Doug Tallamy's lab group, where he and Karen Burkhart went out and they found 12 yards, 6 which were predominantly landscaped with native plants, and 6 which had more traditional landscaping, where maybe they had 1 or 2 native trees, but the majority of the plants were not native. They found that caterpillar abundance was significantly higher in yards landscaped with native plants. And even more impressive to me is that they found that that relationship cascaded up to the 3rd trophic level. More native plants on the 1st trophic level meant more caterpillars on the 2nd trophic level, meant more birds on the 3rd trophic level. And Tallamy and Shropshire on that same year published another study where they did taxonomically comparable comparisons. That's probably redundant, but they did plant pairs of a native versus a non-native in the same taxonomic group. And they looked at the number of butterflies and moths that fed on those plants. And they found, particularly for woody ornamentals, that when you go to native woodies, you get a 14 times increase on the number of Lepidoptera, butterflies and moths that can feed on that plant. Across all plants, there was a 3-time increase when you go from exotic or introduced to native plants. But remember, I told you that plants are not sitting ducks. They pump out all of these chemicals to try to keep insects from feeding on them, but that's not the same relationship with nectar. When a plant flowers and they put out nectar, the sweet little sugar water, just like lemonade or sweet tea on a hot summer day, they're actually trying to invite insects to visit those blossoms because they want the insect to visit so that they can be pollinated. So people who have studied plant nectar found that for the most part, nectar is not chemically defended at all because the plant wants the insect to come and sip the nectar so that it can get pollinated. Furthermore, I didn't think initially that native plant status should matter to pollinating bees because folks who have studied what different bee species do found that the vast majority of them are generalists, capable of foraging from plants across diverse families. 65% of our Western US bee species are estimated to be generalist, where plant family makes no difference on the foraging decisions of that bee. So when Erin Anderson came to me and said, I want to study bee associations with native plants, I sincerely thought, well, that's an awful lot of time and effort to put into a study where you're going to find that it makes no difference whatsoever. But my personal approach to mentoring folks through graduate school is that I let my students pick their project within the context of what I actually have the ability to mentor them on, because graduate school should be about the production of new knowledge. And when you're trying to produce new knowledge, invariably that process is going to be difficult, challenging, or flat out suck at some point in time. So I feel like when the students choose their own project, it helps them stick around for the tougher points. So Erin chose 23 species of Pacific Northwest native plants, and we had 4 non-native comparators. The 4 non-native comparators, including Grasso lavender, we didn't choose them to do direct comparisons. Instead, we chose them because we thought of them as benchmark plants. We knew from our work in the Master Gardener program that gardeners would always come up to us and say, if you want to study a pollinator-friendly plant, you should look at lavender, or you should look at Nepeta, or bees are all over my oregano, or they love my salvia. And so we thought, since gardeners already seem to intuitively know what plants were bee attractive in their own garden, we could tell them, like, hey, you know all the different bees that you see on lavender? Yeah, they're super abundant, but most of them are non-native honeybees or relatively common bumblebees. If we go to Doug Astor, by comparison, Doug Astor is able to support 74 different species of bee, and California poppy and farewell to spring, 44 and 45 species of bee. When Erin first came out with this data for a while, I used to just kind of joke and say, like, we should write a paper called Lavender is a Trash Plant for Bees. But then the more we dug into the data, we realized that yes, you do get a lot of honeybees, you do get a lot of yellow-faced bumblebees, which are very common on lavender, but we also found significant associations between lavender and Bombus viridis and Bombus calignosus, both of which are bees that are IUCN red listed as being vulnerable to, they're currently in decline, and they're vulnerable to becoming endangered. So Erin kind of opened my eyes at like, wow, we really do have some native plants in our area which are super attractive to diverse and abundant bees. Another thing that came out of Erin's work on this project, however, is that we started to realize that the native plant marketplace is kind of the Wild West, for lack of a better word. We would buy plants in their vegetative state, which is normal when you purchase them in spring plant sales, and then they would flower and we'd say, this doesn't look right. That it was either a species that was native to another region, not ours, or it was a cultivar, or it was just flat out misidentified. And so I really started becoming interested in what gardeners actually have available to purchase, which for the most part are native cultivars. Buying a true wild type native plant is actually more difficult than you would think. So here I'm going to segue into Jen Hayes's research. I really want to give her credit for these beautiful slides as well as all the great work that she's done. You all know planting for pollinators is popular. When we surveyed master gardeners many years ago, 93% of the folks who responded to our survey said that they're specifically choosing plants to try to attract more pollinators into their garden. And then there's a big plant native movement, in large part encouraged by Doug Tallamy's work and others. And native plants, we're defining them the same way that the USDA NRCS defines them, as plants that are indigenous and that have evolved and occur naturally in a particular region, ecosystem, and habitat, usually before colonization. Okay, so we know that planting for pollinators using native plants is very popular. However, surveys that have been done show that native plants make up only 13% of nursery plant sales across the United States. And this one particular study that was done in the Mid-Atlantic region in 2015 found that 77% of the native plants that were on store shelves advertised as native were actually native cultivars and not wild-type native plants. How can you tell if a plant is a native cultivar versus a wild-type native plant? Well, one thing you can look for is whether or not there are single quotes and a really fancy or jazzy name behind the genus and species name. So in this case, the lower photo, Doug Aster Sauvy Snow is the cultivar of wild-type Doug Aster. And from many years ago, botanists have been studying how the modification of plants to produce ornamental cultivars may impact insect visitation, and they have suggested that there are many ways that plant breeders can change plants, which can reduce insect abundance and diversity. And current research has kind of been like inconclusive on this topic. There have been several studies in other regions of the United States, none done in the Pacific Northwest until Jen's study, that have looked at insect abundance on native plants versus native cultivars, and they have relatively inconclusive results. As I mentioned, I think that one of the reasons that they had inconclusive results is because they didn't identify their insects to species, but instead it was kind of like honeybees versus all other bees is like two groups of comparison. So having that taxonomic expertise is really important. So Jen set out to see whether or not Pacific Northwest native cultivars, such as this purple gleam, California white, or California Mikado cultivars of California poppy, whether or not they were as attractive to pollinators as the wild type species. She specifically looked at syrphid flies, bees, and butterflies in her study. She also wanted to know whether or not different pollinator groups might respond differently to the cultivation of native plants to produce native cultivars. So for example, many leafcutter bees and many longhorn bees are known to be specialist among the majority of bees, which are generalist. We had three different hypotheses. It could be possible that the native cultivar could actually attract more pollinators than the wild type native. This could be the case if the plant breeder produces a flower that has a highly attractive insect color, such as yellow. Many insects are highly attracted to the color yellow, more so than the color white. It could be that cultivation reduces insect visitation if they change the color to something that insects can't see. So most pollinators can't see in the infrared range of the visible light spectrum. Most bees are thought to not be able to see the color red. Or there could be no difference if the relative color change between the two, for example, is relatively slight and not so drastic that we would expect it to make a difference. So Jen conducted a study for 3 years at the Oak Creek Center for Urban Horticulture, just off of 35th Street on the edge of the OSU campus. She started with 5 different native plant species and 1 to 3 cultivars of each and 5 replicates of each plant, and then planted them out in 1 meter square blocks in 4 rows at Oak Creek. Here were her initial study groups. The wild type native is the larger plant, and the cultivars are smaller and in single quotes. Now, here's where we started to realize that buying a native plant is not easy. We actually knew this from Aaron's study. So Aaron had to go to about 8 to 10 different vendors to get enough native plants to actually do his project. Jen had to do the same. She wanted to include Sadalcia rose checker mallow in her study, but when the 2 cultivars, Party Girl and Perpetta, came up, we were like, man, they look really different than the wild type native plant. So we took it to the OSU herbarium, and Aaron Liston, the curator, told us that rose checker mallow just recently underwent taxonomic revision, and that our native species is Sadalcia asperella, which you cannot buy on the native plant market as far as we know. The only Sadalcia that's on the native plant market is Sadalcia malviflora, which is now native to California in the reclassification. And the 2 cultivars of Sadalcia were made with that California native and not that Oregon native. And so since we wanted to do a study on Oregon native plants, we were like, well, guess we got to throw that one out because it's not going to be testing any associations between native flora and fauna. Calistoga did not overwinter well, so it got thrown out. Nemophila, baby blue eyes, never performed well. We just couldn't get it to germinate well. We couldn't get it to establish. So it was thrown out. And I do want to mention that Camas did well at all of our plots, but it rained so much when Camas was in bloom that we couldn't get good pollinator data because pollinators don't fly when it rains. So those were thrown out. Jen added in novel cultivars, Moonshine to take Calistoga's place, Scarlet, just because honestly, a lot of what we chose was driven by what was available in the native plant market and what we could find. This beautiful purple gleam was added to the California poppy group, so that these were our final group of plants. Remember, once again, you can tell a native cultivar by the single quotes and the fancy name, like Xeratones, Moonshine, Salmon Beauty. When there is an X in front of the cultivar name, that means that it is an interspecific hybrid. Interspecific hybrids are more highly bred in general than other plants which are not crossed across species. So here we have a range of cultivars that are highly bred or interspecific hybrids, and those that are more light selections, for example, Sauvy Sky and Sauvy Snow. When we talked to the breeder about these plants, they said that they were selections of naturally occurring color variants that occur on Sauvy Island. So these two are our less developed cultivars, if you will. They sat for 5 minutes and counted and identified to the lowest taxonomic level all of the bees that visited the plots. They vacuum sampled all of the bees that they could see on a plant after they were done doing the observations. The only exception was for the European honeybee. They just collected 1 voucher specimen and then counted the number of honeybees they saw on each plant because we didn't want to waste like tons of cabinet space for honeybees. There are a lot of honeybees on a lot of these plants. The group was able to identify when they did their abundance counts to honeybees as a group, bumblebees as a group, leafcutter bees as a group, longhorn bees as a group. And I just need to give Jen a shout out. She drew all of these little icons. She's so talented. Syrphid flies as a group, butterflies as a group, and then every other bee that didn't fall into these other bee groups. We also looked at all pollinators as a composite group as well. And there were two main measures we were taking: abundance, the number of individual bees and other pollinators on a plant, and then richness, which is the number of unique species on these different plants. We did statistics. We did more statistics. Okay. So here is the result of the abundance counts. I have no results on here yet. I'm just trying to kind of like prep you for what you're going to see. When there is a green box in a column underneath a cultivar, that means that a pollinator group, either honeybees, bumblebees, all pollinators, etc., had a significant preference for the native over the cultivar. When there is a purple box, that means that whatever row it's in, that pollinator group had a significant preference for the cultivar over the native. And if it stays white, that means there was no significant preference between the two. I'd like to preface this by saying, remember that what we know about bees in general is that 65% of all species are generalist and should have generalist feeding habits. So here are the results. We found that in the vast majority of abundance counts, there was no significant preference of different pollinator groups for one or the other. Like here, leaf cutters had no significant preference across all comparison plants. But when a preference did occur, it was more likely to prefer the wild type native versus the cultivar. There were 8 instances where the pollinators preferred the cultivar over the wild type native. However, I want to point out a couple of things here. 3 of these 8 were from honeybees, which are non-native to this region. The all pollinator preference for Sauvy sky was driven by the high abundance of honeybees on Sauvy sky. That bumblebees also preferred both Sauvy sky and Sauvy snow. And then our other bee composite group, there was preference for Sauvy sky, which I do want to point out, if you remember, Sauvy sky and Sauvy snow were our least developed native plants compared to the others in this group. The one which really threw me for a loop, and I'm still trying to understand, is this other bee preference for Clarkia ammoena scarlet. Remember, I told you that bees are not supposed to be able to see the color red. And so it was startling to me, why would those bees be more abundant on scarlet than the wild type Clarkia? And the only thing we can come up with at this point is that wild type Clarkia, some variants of it, do have these red eye spots, which may serve as a type of nectar guide. So it may be that bees on Clarkia are pre-adapted to be able to see the color red, but that was really just a true surprise to us. Okay, so here are the results for richness, where bee species richness is the number of unique bee species on different plants. Here we're starting to see much more of a preference for the wild type native compared to the native cultivar. In almost every group, there were more bee species, and oftentimes many more bee species on the wild type native compared to the cultivars. The only difference, once again, is with Doug Aster. There were as many bee species on wild type Doug Aster as there was Doug Aster Sauvy sky. And I'll get to part of the reason why that is towards the end of the talk, why we think that is towards the end of the talk. Oh, you know, and then I started getting into, I'm really excited about this data because it's novel and it shows something that I don't think has been shown before. And so we wanted to look at entire bee communities on the different flowering plants to try to understand how they were associating with plants in a garden setting. And so to do that, this is where I'm like, I'm not sure I want to talk through the statistics, but then I also, if I don't talk through the statistics, then I'm just saying, take my word for it. There's a difference. So I think I'll try to do a little bit of both. This is called an NMDS plot, where you shove data in 2 dimensions, and then you try to see if there's any logical grouping between where those points fall out. What we did is we mapped bee points relative to flowers that they could be found on, so that we could look at how closely different bee communities on flowers were in space. The closer they are together, the more similar the communities are. The further apart they are, the more dissimilar they are. Okay. So in the plot I'm going to show, the native plant is going to be a square. The cultivar is going to be a diamond. The different plant groups, whether it's Achillea or yarrow, Aquilegia or columbine, Clarkia, farewell to spring, Escholzia is California poppy, or Symphyotrichum, which is Doug aster. Those are delineated by the colors. And then the bee species are mapped on top of those. So there are going to be a lot of points coming up here. I'm going to try to walk us through a couple of them. So let's look at this purple group right here. In the square is the wild type Doug aster, and the 2 diamonds are the cultivars. These are all clustered fairly close together, letting us know that the cultivation of Sauvy Sky, which, sorry, they renamed Sauvy Star. Sauvy Star and Sauvy Snow didn't really change the bee communities associated with Doug aster. They're very similar. Kind of the same for California poppy. Here is the wild type, the square, and the 3 cultivars are relatively close to that wild type. Things start to get a little bit different when we look at Clarkia. We have Aurora and Dwarf White, kind of a pink and a white cultivar of Clarkia, clustered relatively close. However, it is interesting that Dwarf White Clarkia is actually closer in space to the wild type California poppy. And Scarlet Clarkia over here is more closely related to the wild type columbine than it is the native plant that it was derived from. Oh, and then the other thing I want to point out is that we do have several bee specialists, Megachile gravita, Megachile brevis, Melisodes clarkia. They're closely associated with the wild type native, more so than the native cultivars. Same for Melisodes microstictus and Melisodes lupinus, more closely associated with the wild type plant than the cultivars. Thank you for humoring me with that. If you kind of did not want to listen to that because it was not fun to listen to, I will give you the bottom line. Bee communities, when we look at the entire bee community, they tended to be more strongly associated with the native versus the native cultivar, particularly if they're known to be specialist bees from other studies. Several of the specialists that I talked about are specialists on Clarkia, farewell to spring, and they are actually petal cutting bees as well. We found a lot of petal cuts on our farewell to spring. These photos, I just love them, taken by the undergrads in the lab. A female will excavate a disc off of the petal. Here she is grabbing onto it, just ready to make the final cut. And then, I mean, look at this photo that Mallory Mead captured. She will fly with it and take it back to her nest, where she literally will wrap her brood in little Clarkia petals and raise her brood in a very colorful fashion. So it's a different type of foraging for nest materials off of native plants. If you remember back to that first garden ecology lab brief page that I showed, Mallory Mead showed that there was a significant strong preference for the wild type native over the native cultivars. So it affects not only pollen foraging, but also foraging for nesting materials. When we looked at where our bees were collected, that are known to be specialist. And this is for pollen foraging in particular. And you may know that only females collect pollen so that they can feed it to their babies, that the vast majority of specialist bees were collected from the wild type host. This is causing us to suggest that if you want to provide greater resources for specialist bees, which are oftentimes excluded from garden habitats, you can plant more plants in the Asteraceae, Ericaceae, Malvaceae, or Onagraceae families. And I'll give one shout out for Onagraceae, of which Clarkia is part of that family. When I've collected bees in other studies and we did host plant associations, the new species that were discovered, this was from the New York study that we did, they were all collected off of Onagraceae. And this is just like outside of New York City. We were discovering new species of bees in people's backyard because they had Onagraceae as part of their garden plant palette. I don't feel like talking across a second NMDS plot. Just take my word here. This looks kind of like somebody just threw BBs against the wall. It's very random. With seraphid flies, unlike bees, there were not strong patterns of association with the wild type native versus cultivars. And that's because the seraphid flies are generalist, feeding on nectar, versus bees are more specialist, feeding on pollen that they need to feed their brood. Okay, so we are at the conclusions part. We found all three scenarios of our three hypotheses. We found that there was often no difference in preference for native plants or cultivars, but when a difference occurred, specifically for bees, native plants were more likely to be preferred over cultivars, and they always had a greater bee species richness. We found that specialist and petal cutting bees were more likely to be associated with native plants than their cultivars. We found that the cultivars that were consistently preferred over the native plant were in the Doug Astor group and included some of the least developed or least bred plants in our study. A little bit more about what's going on with the Doug Astor group. Here's Savea at Olify 4, standing next to the native Doug Astor plots. And here she is standing next to Sauvy Snow and standing next to Sauvy Sky. I'll mention that we did not correct bee abundance or richness counts by petal number because we figured like, you're not going to do that. You're not going to even up all the flowers in your garden. And we thought it was a good representation of what actually might occur in someone's garden. When I was visiting the field site one day, Jen yelled across the field, like, don't water Sauvy Sky. We don't want to give it any more power. You can see that it grows like twice the height of the wild type native. When I've talked to other gardeners about these plants, they said, well, as a gardener, I would never let it get that big. I would shear it down halfway through the growing season and then shear it down again. So maybe that isn't the practice you would use, is to let it get as big. But part of the reason we think that it attracted more bees is just because it had more blossoms. Ultimately, we recommend, if possible, selecting for wild type native plants to support the greatest abundance and diversity of pollinators. If you do choose cultivars, seek out near native or less developed cultivars if possible. And I just want to note that Jen has looked at all of the different traits that might be able to explain the patterns that she's seeing. She's taken corolla width, corolla depth, plant height, plant width. We bought this thing that you can point at a flower, you can point at paint, you can point at a shirt, and it'll give you the hex color of that plant. So we're trying to look at like color differences between the wild type native and the cultivars. We've also taken photos of the plants under UV light. This is a really neat photo. California poppy, you may or may not know, doesn't provide a nectar reward for bees, only a pollen reward. And here, it's hard to see in this room, but it's the pollen that's fluorescing the UV on the wild type native plant. And when we look at the same cultivars under UV light, the pollen has lost its fluorescence. So we looked at nectar content. We looked at sugar content of nectar, really found no difference there. We collected pollen off of all the plants, which was a real bear to do for something like yarrow. We sent it off to get analyzed for amino acids and sterols to look at nutritional content. Honestly, we think that right now color is driving the bus here. It'll be interesting to see what comes out with the pollen data. So with that, I thank you for your time and attention, and would be happy to take a question or two if there's time for questions. Yes, hi Janet.
So listening to you, I want to go buy plants or seeds that are.
Yeah, where do you buy seeds or plants that are Oregon natives? Where do you get them? I would work with native plant growers and through your local native plant society, the local MPSO. That growing native plants really has been a labor of love to the growers who've been doing it for decades, and they have like a real ethos about what it means to grow a native plant and to sell a native plant. Now that native plants have become really popular in the marketplace, there are new players that are entering the marketplace, and it's harder to tell. You can tell if there is a single quote and a fancy name like Party Girl, then you know that that's a cultivar. But there's also like some of the things that I've seen in the native plant marketplace is that there's a trend towards calling things a North American native. So it's native to the continent, might not be native to your region, but it's native somewhere. Everything's native to somewhere. So I think honestly, working with local growers who you know, working with the Native Plant Society of Oregon, who I've heard the Native Plant Societies described as, not the philosophical, but kind of like the conscience of the native plant movement. Like they have a real strong ethos about what it means to collect, grow, and sell native plants. Hello.
So Sylvia Island natives, I think they do a lot with the Portland group.
So the Island Natives works a lot with the Native Plant Society of Oregon and the Portland group. Yes, they do. They are big native plant growers. They also do have some cultivars for sale. They are the originators of the Salvi Sky and the Salvi Snow. Hello.
That's a nice question, but I go medicinal herbs, and the bone set that I received from an oak teacher has the most variety of insects on it I have ever seen, and it's very, I like to start lighting the garden every time. Are these already known generally, or is getting the word out that, correct me if the bone set isn't a real attractive diversity just from my own exposure?
Right, so the comment is that bone set in the garden attracts a high diversity of bees. Does anyone here know the genus of bone set?
Just looked it up, and it's Eupatorium purpureum.
Oh, Eupatorium. So thank you for sharing that observation. And then the question was, are these associations of what's highly bee attractive in terms of abundance and or richness? Is this well known? And it isn't, because the research that I've shown you tonight took a collective like 7 to 8 years to do. And it takes a lot of time and energy. And so sometimes that's part of the reason why we're starting to look more at plant traits, because I'm like, do we have to test every plant? Like, no one's going to test every plant. So like a lot of the research hasn't been done on most plants. And so we're trying to come up with a more predictive model that can help people understand what's likely to be a pollinator attractive versus non-attractive plant. Of course, that's already been done in the 70s through like pollination syndromes, people coming up with that rubric. However, the more we study plant-insect interactions, we know that pollinator syndromes are not perfect or even in many cases good predictors of what will be on different plants. Like, for example, it makes no sense that we have an abundance of those other bee groups on the scarlet clarkia. They shouldn't be able to see red. So I don't know off the top of my head of data that exists for bone set, but that just could be because I don't know.
I guess I was also wondering if this was the anecdotal information is useful.
The question is the anecdotal information useful? And the anecdotal information is fantastically useful for helping us to come up with future studies that we'll focus on. And then Judy, thank you for sharing that the Oregon Flora website has a list of some native plant nurseries. I'll say something about native plant nurseries though, in terms of like, we did a survey of all native plant nurseries in the United States for another grant I was working on. We started with the list that was given to us by Rick Martinson, who did his PhD project on native plant nurseries. In the 3 years between when he gave us the list and when we finished calling every nursery we could in the United States, about 16 to 20% on the original list had gone out of business. And like we doubled the list, more than doubled the list from 329 original nurseries to over 800 native plant nurseries serving ornamental marketplaces. So I'll say that with my colleagues, we talk all the time about like this list. Where's this list of native plant nurseries going to come from? Because it's really dynamic and it's hard to keep up to date. That's why, once again, I think working with your MPSO chapters, they're really great resources. They know what's going on. Thank you, folks. Oh, sorry, one more.
So do we know that these, I'll just say these, these not pollinators, are primarily attracted to plants because of their pollen, or can we rule out that there's I'm using the word smell, I don't know what else to use.
Right. So the question was, do we know that bees are primarily attracted to plants because of their color, or are they also attracted by the smell? Most biologists think that it's a two-phase recognition process, that from further away, it is color, and not only color, but the contrast of color against the background, which can catch the eyes of bees. And that as the bees get closer, then that's when the chemical sensations, the smell and the taste can play a role as to whether or not it's acceptable or not acceptable as a forage plant. So
I did wonder if some of those characteristics are bred out of the plant or minimized as the cultivars are developed. Just like roses often become, I think, I'm not a rose specialist, but
Right.
become less fragrant.
right. Yeah, so originally the question was is whether or not some of those chemical characteristics or more subtle characteristics get bred out of the plant as you're selecting for larger petal size. Like the one that we found is honestly the ultraviolet signals, like we wouldn't notice that, but those are changed quite heavily during the breeding process. I had hypothesized that nectar volume and sugar concentration would change because you're selecting for larger petal size, and it's kind of like a water balloon. When you squeeze from some place, it like contracts in another, but we did not find any difference that we could discern in nectar volume, nectar volume or sugar content between the wild type natives and the native plants. So thank you folks for joining me this evening. I'll stop recording. Thank you, everyone.